Hi, Tom. Can you tell us a bit about the type of problem that you're interested in?
So I'm interested in finding effects in data, which is usually data from the brain. And so this is data that takes the form of a spatial process. And we're looking for effects. These are differences between groups of populations. Either it might be healthy populations and people with disease, say schizophrenia, or it could even be within a group of healthy subjects. And we're looking to see where are there effects that can be explained by a genetic variant or some characteristics of their environment.
Can you tell me a bit about why that's a difficult problem?
So the data that comes from brain imaging is difficult in two ways. One is that it has spatial covariance, so it has a spatial structure that is unlike the normal statistical assumptions. Usually we assume there's no dependence between things we measure. The other thing that makes it complicated is that when we measure multiple measurements per subject over time, there's also dependence. So we say there's spatial dependence and there's temporal dependence, and both of these are sort of complex. And to add on top of that, that's just the noise, also the spatial structure of the signal. The temporal and spatial structure is also different and complicated. So we have to sort of grapple with that in order to get inferences that we can trust.
And can you give us a sense of your approach to solving these problems?
Sure. I've actually sort of taken two approaches, and I would say they're kind of low-tech and high-tech. So low-tech are using methods which make very few assumptions about the data, and then they use computational methods to basically come up with inferences that we can trust. So these are sometimes referred to as resampling or permutation-based approaches to the data. And this is very good because we know we can trust the results because they're making very few assumptions. However, as the data gets larger and very interested in big data, these things may not scale so well. And so I'm interested in both methods for speeding those methods up, but also other methods that make stronger assumptions and can deliver us those same inferences using sort of sophisticated mathematical techniques that tell us about how the structure of the data.
And can you give us a couple of examples of either diseases or problems in biology that you're trying to solve using these approaches?
Some of the methods I've been working on have been applied to multiple sclerosis. So multiple sclerosis is a disease that is actually very dependent on magnetic resonance imaging for both diagnosing and tracking the disease. So we're in need of methods that will characterize the degree of the lesions, these are the deficits in the white matter of the brain, we need to be able to track them over time and see how they change, and then see if they relate to different aspects of the disease. For example, is someone suffering more from upper body deficits as opposed to deficits in balance or what have you?
And you mentioned the excitement about using big data to try and probe these kinds of questions. In the case of multiple sclerosis, for example, how do you see big data helping us tease apart what's going on in terms of the disease processes?
So for big data, getting big data on populations of patients can be challenging, but we're always trying to press the N. But with the UK Biobank, we have an enormous resource of up to, we're passing 20,000 subjects, and soon we'll have 100,000 subjects of images of typical white matter. So we'll be able to develop more accurate models of how do white matter lesions occur naturally. So it turns out that they're slightly different, but there are types of lesions that occur in white matter in healthy subjects. All of us, as we get old, will likely have some of these white matter hyperintensities. And as we are able to better develop models for those sorts of lesions, we should be able to apply that methodology to help us better track multiple sclerosis patients.
And what do you see as some of the big challenges or the problems that we face in trying to make sense of the population scale data that we might get from, say, the UK Biobank?
I'd say the challenges are both, I would say, computational, statistical, and interpretive. So computational is kind of what you might expect, just how do we scale up these methods, and I think there's a lot of interesting ways to do that, and I'm very excited to work on that. Statistical is how do we accurately model this data that traditionally we've been using methods that were intended for 10, 20, maybe even 100 samples, and now we're applying them with 100,000 samples. And it turns out some of those methods basically break down. For example, null hypothesis testing is something that if you throw enough data at it, it will just always say there is something there, which is not very useful. And then the interpretive is where I think we need to work with our colleagues in epidemiology to really get a grapple with the confounds, because even if the best designed statistical methods finds an effect, it could be that that effect is due to something that we're not aware of. It could be an environmental difference or some sort of confound that we have not measured. And so that, I think, is layered on top of everything else that we do, is the interpretation.
And then finally, give us an example of something that you'd really like to achieve over the next couple of years.
What I'd really like to do is develop methods that are going to work on the UK Biobank at scale, and see those methods be developed into tools that are used around the world. So right now we have tools that are used commonly, and I've helped develop some of them, and they are used widely, but again, they're developed for hundreds of subjects and what have you. So I think there's a great need, as more and more sites around the world develop these biobank scale data sets, seeing that work developed and shared.
Thank you very much.